All right. So right here is the first one that we are reviewing for the question. Just very straightforward, no issues as such. We will just give you that, say for example, which of the following are the properties of the transformed cell, and some of these will be in there, and that's what it is. Or sometimes what can happen that we can just give you something like which of the following is not a property of the transformed cells, and we can just change one of these choices to a negative one. So say for example, I'm just saying that it's immortal can go indefinitely, and I can just change with a not, which of the not following, that mortal can grow for certain generations, something like that. Or say they will lose growth upon contact. So just the other way. So very straightforward question. You will be able to get it. All right. So after that, we are stopping at. All right. So we are jumping a number of slides, and we are stopping at the retroviruses. And this one is basically the difference between the Rous sarcoma virus and the ALV. So as I just put right here, know the differences as it is in the slide. And one of the important thing that just remember that what, if you just think about that, what are the retroviruses, some of the knowledge of the previous chapters are required. Remember that all the retroviruses have a plus sense RNA, and many of them have a diploid genome. That means that identical two plus sense RNA have reverse transcriptase. So just know these basic things which are added on top of that as such. And let's see. So one of the most important part that we are just going to see in the next couple of slides, that what is the major differences in between this Rous sarcoma virus and the avian leukosis virus. The major difference is that all of these functional, which is a cancer-causing recombinant ALV, have a different host DNA, and on the other hand, what happens that all functional cancer-causing RSV, they have the VSERC or the viral SERC gene inserted in there. So that's what it is. Very different and very different. And so that's one of the major differences in between them. And here it actually shows right here. So rapid tumor formation with the RSV, not as a separate question, but just the extension of that. RSV has activated the dominant oncogene in the VSERC, and protein produced immediately when the virus replicates, that's basically an oncoprotein. And here, this is for the ALV. I should have highlighted this one as well. No dominant V-onc gene, and it's basically the cis activation, expression of endogenous oncogenes. That's basically the process. So here also, it says that if you just take a look at the Rous sarcoma genome, you have the GAG-POL envelope that is present in all retroviruses. Right here is the VSERC. So you can easily see that along with the promoters, it's inserted in there. But if you just look at all the different avian myeloblastosis virus or the other one, most of them are defective, where you can see a portion of these GAG-POL envelope is compromised, and all of them are different. And that's the reason that they need a helper virus to produce the virus, and as they have all different things, their activation process is different compared to that. So that's the major difference in between them. So just to know that, so it should be all right. So basically, just remember that there are a couple of things but the question basically will ask that here you can see that there are initial viremia, older birds develop leukemia, sporadic, and a couple of different things that both of them are plus sense RNA viruses. I have already said that they're retroviruses. And Both of them can transform cells. Remember that both of them are retroviruses, cancer causing, so both of them transform cells. So these are not the differences, not the major differences as such, but the way I said that the VSARC is inserted in every RSV genome, whereas it is not in the case of the ALV. All right, so that's all good. Then let's take a look all the way we are talking about right here. All right, so just the basic process that how, as the slide says, that what is basically the mechanism or the process used by the DNA tumor viruses to transform the cells. And you are seeing that basically the T antigens kick the quiescent cells into the S phase. This is one of them. They inactivate the p53, and the details are in there. So these are some of the one. And remember that this is basically the idea that in the cell cycle, this is your growth 1, G1 phase. Then you have the synthesis phase, and right here in the G1 phase, you have the restriction point or the restriction point. So what happens that the cell can be arrested at the restriction point, and when these T antigen basically binds the cells, even if their cell is not ready for scheduled DNA synthesis, they will undergo the synthesis for uncontrolled division. So just remember that it binds to, the T antigen binds to a protein which is responsible for the restriction point arrest. And you know that very well, that one of them is RB, that it just holds the cells in there, and these T antigens kick them. And what we are going to see that the T antigens is of a tumor antigen basically is a generic name. We will going to see that there are very specific ones. And for SV40, for HPV 16/18 human papillomavirus, as well as we have that for the adenoviruses. This is a generic name, but they are all different proteins. We are going to see that in a second. So right here, there is the little bit of detail that you did not have in your original slide. And what we are looking here, that we can ask that which of the following are used by, say, for example, E1A, which is the T antigen, SV40LT, that's T antigen, or HPV-E7, that any of these proteins, what is the mechanism by which it can arrest, or which of the following is not a process to arrest the cell cycle. So right here in the specific detail that what these protein does, here I just mentioned adenovirus, but all of them do the same thing. And so they bind to the RB, and basically what happens, they removes the HDACs to allow the transcription of the genes necessary for the DNA synthesis. So when you recruit the histone deacetylases, that's basically those are responsible for condensation of the DNA. They remove those. And then bind to the RB, free the E2F and the DP, the dimerization partner. So RB, E2F, DP, and HDAC. This complex is basically arresting the cell division. So when E1A binds to this RB, it replaces, you can easily see, the E2F and the DP, and the cell cycle can go on. And also along with that, this is not from this slide. This is a different slide, but I just put it in there so that you can make a connection. It also, the E1A, bind to the P53 followed by ubiquitination and proteasomal degradation and allowing unscheduled DNA synthesis. So this is from a different slide, but I just put it in there because we can ask the functions of the E1A like this. So just to make sure that, so if I given the function, all of them, if they are there, your question is basically that all of them are the functions for E1A. All right. So that's all good. Then we are going into this slide, which is a busy slide. So here I just added a couple of different things that what we have here. You have the E1B, E4, also along with that E1A, which we did not talk about, and then HPV 16/18, E6. So remember that it is human papillomavirus type 16, human papillomavirus type 18, and the protein is E6. and SV40LT. So all of them are these T antigens which promote the generic name is T antigen because all of them basically kick the cells into division. And see that what are the different ones. I'm going to just read this and show you that what is happening. The first one is the sequestration of the p53 and abolish its DNA binding ability for transcriptional activation by SV40LT or large T antigen. So right here you are looking at this. So it binds to the, right here is the LT and the p53 is a tetramer. And when it binds, it cannot do its function. It cannot bind to the DNA. So this is sequestration. Then suppression of p53 mediated transcriptional activation of apoptotic cell cycle arrest genes by E1B. So basically E1B right here, this is what we are talking about. E1B binds to the p53 and that will basically block the apoptosis right here. You are looking at this portion connected to this. And the next one is the ubiquitination followed by proteasomal degradation of p53 by HPV16 or HPV18 E6 protein and adenovirus E4 ORF and E1B protein. So again, the names will be given to you. It's the function that is important. So what we are looking at here, we are looking at adenovirus E4 ORF6 and E1B. Here we are looking at HPV16 or HPV18 E6. Both of them, if you just look at, you do not need these individual names, but what they are basically doing, right here is the E1B and right here is the P53 ORF. Right here is the E6. All of them are binding to this purple P53 and recruiting this big complex right here. And basically these big complexes are ubiquitin ligases, which basically puts the ubiquitin onto the P53 and they are degraded proteasomally. Remember that ubiquitination is the universal signal for protein degradation. If you label any protein in the cell with this ubiquitination or ubiquitins rather, they will be undergone degradation. So definitely remember this one. All right, so let's see right here. Not much. So that's pretty quick. So right here, a very basic idea about that which of the following is proven to be the best defense against the acute viral disease. And if we just give you different things like antibiotics. Remember that antibiotics are always against the bacteria. Antibiotics has nothing to do is antivirals, but that's not the best way to do it. And also, if we say natural immunity, passive immunity, those are secondary, but the vaccines basically are our proven best defense against viral defense. So just the heading of the slide could be asked that what is the best defense against, because the reason is that if we just ask antivirals, they are much less. Most of them don't work as best as the vaccine. So it's still the best against the disease. All right, so right here in this question, this is a natural passive immunity. And the question is that how a newborn child remain healthy before the first vaccination are administered on the onset of a well-developed or the onset of the well-developed immune system. Remember that it at least takes 6 months for the infant to start getting those IgM, IgG, or IgA level. Remember that right here at birth, the infant does have the mother's antibody in a large amount, which the IgG can cross the placenta and in the circulation. And after birth, the antibodies are supplied through the breast milk, and antibodies are not, they are glycosylated in such a way that they are resistant to proteolytic digestion. They can cross, we have seen that before, that detail is not necessary, but they can cross the intestinal lining and by a process called transcytosis and get into the circulation. And so basically natural passive vaccination or the antibodies from the mother to infant basically protect them. So very straightforward, nothing complex. All right, so here let's see. So here what we are looking at right here is the humanized antibodies. Remember that. The chimeric antibodies, the part human and the part from the mouse. whereas the humanized antibodies are almost all human. Only the scaffolding, which is the complementary determining region, those are against the antigen. So what we are looking at here, again, you do not have to go into the technicality of that, but just remember that this is basically the humanized, which is the murine CDR sequences, which is called the CDR grafting. And this is where the mouse antibody basically recognizes the epitopes of the antigen. And the rest of the part, all the scaffolding, is the human. And this is the thing, that the humanized mouse monoclonal antibodies are much less immunogenic. So that's the reason that you can see that this is immunogenicity, which is the highest in here and is declining. Of course, there will be nothing human to human. And here it is declining. So this is the advantage of the, so if you're just being asked that what is the advantage of this and what is the reason for that, because remember that a mouse antibody against a antigen or a virus, if you purify that and make a hybridoma out of that and inject to the human being, all these portions are antigenic or a foreign antigen because it's from a mouse to the human being. So immediately your immune system is going to attack these parts. Whereas here, as they are the human type, the immunogenicity is much, much less and well tolerated. And so all the monoclonal antibodies are based on these humanized monoclonal antibodies to these days. All right. So here, just the different types of vaccine that what we have, we will just ask is like which of the following ways you can make vaccines against a virus. So you can easily see that you can make recombinant vaccines, attenuated vaccines, inactive vaccine, make subunit vaccine. So just the different types, very straightforward. And if they're given multiple times, like multiple correct answer, all of them could be the answer as well. All right, so after that, we did not spend much time during the class in here, but these are just some very straightforward informational slides. So just remember those slides. And we can just ask is that, say for example, which of the following statement is correct about Edward Jenner's cowpox vaccination? So one of the things that this was already in the notes, but the vaccine, the word comes from the vacca, that's cow in Latin. So that's one of the things. And these are some of the observations that the milkmaids get the cowpox rashes in their hands, but they are immune to the smallpox. And the cowpox virus was similar to smallpox virus and provided the protective immunity. So let's see. Yeah, so that's one of the things. And another thing that I can just add, that basically what happens, that the cowpox vaccine basically caused a local infection, but protected against the dangerous smallpox virus, which is basically, you can see that primary vaccination site, you can have a local infection, and that's why you can have a scar, but nothing compared to the disease. So that's basically what is. It's just the basic idea that about the vaccination, that what are the correct ones for this purpose. So just know that it was a cowpox virus, just the principle of that, and that it can cause a local dermal infection of the cowpox, but that is good enough to protect against smallpox. All right, so after that we are going to the herd immunity. One of them is just the definition is that one of the important thing is the key concept is that, remember that once the herd immunity is achieved, at that time, what it says that this is the most important line, I just did not put it in here, it's in your notes as well, that it, the thing is that basically if we just ask, is that herd immunity basically emphasize what? Is that not everyone in the population need to be vaccinated as such. But once a threshold number, which you call, is reached, then apparently the virus doesn't find its way to infect a new host and gradually disappear. So that's the important thing, that herd immunity does not mean that 100% of the population needs to be vaccinated. It depends upon the viral, how it transmits depend upon that. So that's one of the things. And the important part is that just remember that simple thing. So you can have a blank paper. Just make sure that you show the blank paper to the camera. And you can just give in a simple arithmetic like as simple as this. You do not need any calculator or anything like that. But remember that in your lockdown browser, you can access the calculator if you want to. But you may have to just compute one of the herd immunity. We will give you these data just like right here, the R naught data, not necessarily the same value, but something similar. And you may have to just compute, just do this simple calculation. It is basically the herd immunity is 1 minus 1 over R naught. So it's super, super easy to do it. All right. All right, so after that, let's see where we stop. This is the same thing that we talked about this a couple of slides back, that what are the different types of vaccine, how you can make that. So know that you can make an attenuated vaccine, which is alive. You can make an inactivated vaccine. You can do the fractionation. You can just make the replication competent vector vaccine. Protein fraction are produced in a heterologous system. That means that proteins you can produce, the antigenic protein of the virion you can produce in a bacterial cell or the yeast cell, and you can do that. And of course, now we have the mRNA. So just the different types, what they are, that will do it. All right. So after that, after that is the adjuvant. So that this is a very important, important that this is needed for the most of the injectable vaccines. And these stimulate the early process, and just know that what are the reason for that, produce a more robust acquired immune response with less antigen, and slow release of the antigen as a site of inoculation. That happens sometimes. They are with the aluminum aluminum hydroxide and or phosphate in the HPV vaccine. You can see what they are. So just to know that one of these, this is a monophospholipid A, a squalene, and sometimes you can just use like which of the following are adjuvants. A couple of these names in there, and these are the adjuvants, and we can just also ask these. So remember some of these names as well. So, one of them important is the alum right here, and then you have the squalene and monophospholipid A and saponin right here. You can underline this. So I'm just specifically telling you to underline the alum right here and this saponin right here. That is important, and the squalene, which is right here, squalene oil water material, and monophospholipid A. These 4 things, these are important in there. These will be given as an answer choice. You do not have to, and if they are given, is like which of the following are the adjuvants. All of them are if you see these names. So just these are just like a Bloom's level 1 recap type of questions, very straightforward, easy. All right, so right here is so here we can just ask a question like that, which is one of the most important or which one of the is a key for a successful inactivated viral vaccine. The main function is that upon inactivation, the virus will completely lose its infectivity, but it will retain its antigenicity. So infectivity is completely eliminated. It cannot cause any disease, and antigenicity is not compromised. So basically what happens that these are all antigenic epitopes which can be recognized by the antibodies, or against this the antibodies will bind, and those will be clonally expanded. So these proteins on the surface, no matter what, whether it's enveloped or non-enveloped, they must be retained. Antigenicity is not compromised. So these are the 2 things which is the key. The second line is more important. All right. All right, so here is one of the slides that we can just ask that which are the drawbacks, where the drawbacks of the Jonas Salk's vaccine, which did not stop the transmission of the polio. So just as I said, that remember that the inactivated vaccine were injected and the poliovirus replicated in the intestine, which can pass and transmit to the other. So the vaccine could prevent the virus from replicating in the intestinal mucosa to stop transmission of the poliovirus, and suffered from the reversion of mutation to cause the paralysis in a few children. So basically, hold on 1 second. This is a typo error. Could not. Very important word. If you miss that, it just says completely. The vaccine could not prevent the virus from replicating in the intestinal mucosa and stop transmission of the poliovirus. So it would have been a disaster if you just missed that could not. It could not cross the blood-brain barrier to infect, but that's fine, but that's the most important part. So just make sure that you make that correction in the slide. So these are some of the things that if you are in the class and watch this is important. The vaccine could not prevent the virus from replicating in the intestinal mucosa and stop transmission. That's the drawback. And some of these basically suffered from the reversion of the mutation to cause paralysis in a few children, so which transmitted to the other people as well. So these were some of the drawbacks. All right. So let's here just informational purpose, not a direct question. Remember that the varicella zoster vaccine, which is known as which is basically caused the shingles. The vaccine is called Shingrix. Remember that it's a Shingrix from the shingles. Just passively could be a distractor in a different question, but directly no direct question in here. So again, this is your basically you are infected with a chicken pox. That's varicella zoster. It just harbors infinitely in your peripheral nervous system, and Shingrix is the shingles vaccine for those. So this is what, on the other hand, more important here, HPV vaccine. So HPV vaccine, there are many different types, and I have just added these things on this side that we can just ask is that which of the following is basically true or not true, as you can see. So the first one of them is that they are secretory antibodies are produced against the vaccine, which protects the mucosal membrane of the genitourinary tract, neutralizing an incoming virus. So right here, this is what is being shown, that these are the mucosal lining where it is produced. And the way it is produced in that, the virus, the vaccine is produced like a virus-like particles from the HPV L1 protein. So you can see that this is a L1, this is a major capsid protein of the HPV. So these are cloned, and these are cloned in different ways. So one of the way that the GlaxoSmithKline, they have a ceravix, what they use, that they use these baculovirus vectors. So these baculovirus are insect viruses which are engineered to produce this L1 protein in the insect cells. So they're produced in the insect cells, see that L1 recombinant, yeast or baculovirus. So that's basically produced in the culture, and you purify that. On the other hand, say Gardasil, there are several types. There is a Gardasil and the Gardasil 9 that's in right here, and against the different types. They are, on the other hand, produced in Saccharomyces cerevisiae. The Saccharomyces, remember that this is an yeast cell. A lot of people actually is given yeast. If this is not said, sometimes people forgot to make that connection. This is your basic biology connection. So Saccharomyces cerevisiae is the yeast, and you can produce these VLPs, or the virus-like particles. Remember that this is a completely empty capsid. They just self-assemble upon production. There is nothing inside. There is no genome, and it's a very, very good vaccine. But cost is high because you see that you are using insect cells or the yeast, so pretty expensive vaccine. All right, so let's take a look here. All right, so here is the Dengvaxia, which did have some of the drawback. And the first thing is that how it was produced. For that, we are just going to this one is right here. So right here, I should have put a star on this one as well. So this is building on the success of a YF17D vaccine. This is an yellow fever vaccine. What they did is that right here is the linear representation of the yellow fever virus polyprotein that is being produced. And what it was done that these PRM and the E, this were replaced right here. You can see that right here. This is the membrane protein and the envelope protein right here. This membrane protein, PR means like pre-M, that needed to be processed to the mature M. But they are replaced by the dengue virus. So that's what it is the EPRM of dengue virus 1, 2, 3, 4 in the YF17D backbone. So 4 different types. On the other hand, what happened, but still the problem, although this is correct, there is no protection against the type 2. And in some of the cases, it led to the worst disease in about 2 to 9 years old. So that was the reason, the drawbacks of the Dengvaxia. It's not licensed in the United States. This is the one which is much better. But all right, so here is the associated problems that with the Zika virus. Very straightforward recap type question. Just remember that in adults, it can have this Guillain-Barré syndrome. Also, along with that, you can have encephalopathy or meningoencephalopathy, cephalitis. This is basically the inflammation of the brain or and inflammation of the meninges or the coverings of the central nervous system. Basically, the dura mater, pia mater, and the arachnoid mater. Those are the 3 meninges that covers. That's not needed, but just saying. On the other hand, for the infants, it's microcephaly and macular or atrophy. These are basically degeneration of macula that can also happen. So just know that what are the things, all of them are important. All right, so here is just a small thing that I talked about that, but just to make sure that you remember that this is a virus, which is a virus vector vaccine. Just very simple that we now know that vesicular stomatitis VSV vaccine is a vaccine for the Ebola virus already licensed, and then this adenovirus vaccine that is produced by AstraZeneca and a couple of other different countries. Those are a vectored vaccine, and there are many. There are probably 11, a dozen of them as a veterinary vaccine in the market. But so basically what the idea is, you have the virulent parental virus, and basically you clone these proteins which are on the surface, either capsid or on the envelope, and put it on the virus, recombinant virus, and that's basically the idea. And in this process, what you are doing, that these are known as the pseudotyped virus. Remember that. So this word is important, that what is a pseudotyped virus is where you change the surface glycoproteins or the proteins or the surface glycoproteins of a different virus. So just the reason for that, what it is. So here is the example of that. See right here. You have, again, not a direct question, but just this, what is a pseudotyped virus? This is a vesicular stomatitis virus with a G glycoprotein. This has been replaced right here with the envelope protein of the Zika. Now you have this recombinant, which is with this. And the VSV is an animal virus. It doesn't really infect and cause any toxicity in the human beings. That's why it can be safely used as well. All right. So here we are going to give you a couple of questions about the mRNA vaccines, and we can just ask about some of the synthetic process that what are involved in producing these mRNA vaccine. So here are some of the most important polymerases that are used in the industry to produce this. And Here you have a T3, T7, and sp6 RNA polymerases. Remember that these are all from bacteriophage, and these are all cloned in the bacteria, and in the industry they are producing this 500 liter, 1,000 liter, 2,000 liter fermenter tanks and produce from there. So basically the industry, when they're making these mRNAs, they literally, I mean, like it's kind of a mind-boggling that they probably have used liters of these enzymes produced artificially in cloned bacterial cell culture and purified. So this is the RNA polymerase that you used for making this. And a couple of other properties that what you have, that both of these, Moderna and the Pfizer, they contain a modified nucleoside, which is the pseudouridine to evade the cellular degradation pathway. So that's one of the important part. And the reason is that what happens is the pseudouridine is perfectly recognized by the ribosome, and there is no problem of the synthesis of the protein. It will be recognized as an EU. But the TLRs, first of all, the mRNA that is being delivered is very, very similar to the cellular mRNAs. They are polyadenylated, they are capped, but sometimes the cell is extremely sensitive and understand which is its own and which is not. But if they contain this modified nucleoside, the detection is a little bit less, so that their half-life is much more, so that they can make much more antigens out of that by translation. And also just the idea that transcribed RNAs from the spike protein gene DNA template are capped, polyadenylated to form the translation-ready mature mRNAs packaged in a phospholipid bilayer in a lipid nanoparticle. So this is basically from the last part that what we are doing in the process, in here. So this is just, again, each step is not required, but just overview that what we are doing. First, we are making the RNA. Then we are capping it. This is a 5' cap. And we are polyetailing it. So that's basically in vitro transcription by these enzymes, capping it, polyadenylating it, and then we are putting them in this lipid nanoparticle, which is a phospholipid bilayer. Remember that it is a phospholipid bilayer. By chance, if I just make this word a phosphoprotein bilayer, it does not make any sense. So phosphoprotein is nothing, or say phosphopolysaccharide. Just looking at the word phospho, don't choose it. So it has to be a phospholipid bilayer. It's a lipid nanoparticle. There cannot be anything other than lipid in there. So just make sure that you probably saw that question in your quiz, but we'll see in your question in your quiz, or have seen in the quiz. So that's basically what it is. All right, so here also we talked about this. Right here is the modified base, which is the pseudouridine, and stability and cellular detection by the TLRs. We already see this. Remember that these are the lipids which are present in there, and these are the ones which make those phospholipid bilayers. So that's basically the idea. Others, you do not have to, no question is specific from here, but this just shows that nucleoside modified RNA, that's what basically I said in the last two slides. So basically from all these three slides is just one question. All right, how much time we have? Okay, yeah, we are good. All righty. So here, one of the very important aspect or question is that why we do have so few antivirals, and it's very straightforward. Every step of the viral life cycle engages the host function. So high degree of safety or selective toxicity towards the virus is required, and it must be extremely potent. So that is the main problem that you can underline, that to achieve the selective toxicity where the virus will preferentially will be killed by some process without harming the cell is the most difficult part to achieve, because most of the viruses use the cellular machinery. That's one of the is a problem. So again, just if you have to underline just only one thing out of the whole thing, that remember that the selective toxicity is very difficult to achieve. For that is the main impediment that why there are so few antivirals. For antibiotics, it's not a problem because bacteria is self-living. They have all different types, similar, but they have all different types of proteins that doesn't, and you can just develop, they have DNA, RNA polymerases and all those kind of things, but they are different than the mammalian ones or the eukaryotic DNA, RNA polymerases, and you can target them as well. And there are so many different targets like the cell wall and everything. Here, even the envelope of the virus is derived from the plasma membrane. So not much you can do. Oh, somehow I did not label this one. I should put a, sometimes, sorry about that. This is also, just know that what is basically, all right, there you go. Yeah, so this one should be there. So we can just ask is basically what happens is that just the definition that if we just ask is the process of dust discovery where you take the random natural products, right here, I will just update the slide, where you take random natural products and screen them from the soil. What is it called? It is the blind screening. So just the second line basically is the definition of blind screening. That will do it. All right. So right here, this is basically the idea of the cell-based assay. So basically, we can just give you a couple of different types that which of the following process is a cell-based drug screening and give you different scenarios. So right here, this is what you need to know from this is that in a bacterial cell, viral, so as an example, I mean like we'll give you the specific example. There are many cell-based assays, but just one of the process that we can just give you an example, that a viral protease cleavage site was introduced in a cellular protein. So see that this is a cellular protein and you have an engineered protease site. And where the cleavage leads to a loss of function, a cleavage leads to a loss of function and to a tetracycline pump basically, right here what you are seeing. And this can be assayed in the presence of an inhibitor to restore the function. So if you just add the inhibitor, then this cannot be cleaved. The protease cannot cleave that one, and that is basically the functional assay. So this is a cell-based screen. Just remember that. All right. So right here also that how you do this same way that we can ask that which of the following is name of the screening, that we can just describe that. That if you just say that is a small organic molecules predicted to bind different pockets on surface of the proteins, different pockets on the surface of the proteins can be grouped as subset of distinctive chemical structures. So these are the subset of distinct chemical structures which can be linked by different linkers using an automated process to screen a very large data set, usually by high throughput method. So you can see that this is a linkers. These are the subset of the different things. You can just bind those. And this process, what you need to underline, is known as this combinatorial chemistry. So antiviral compound screening by the process of combinatorial chemistry basically does this process. So these two things which is highlighted is important. All right, so here we are asking about a specific mechanism that how specifically the symmetrical or the amantadine actually work. Sometimes some of these questions like this could be a little bit difficult. So in the sense that if there could be similar type that I am describing right here, but it may not be correct. So sometimes if none of them are correct, could be an answer, and feel free to choose that. So you need to understand that what's going on. So here, the influenza virus is taken in the endosome, and the vacuolar ATPases are present on the membrane, which pumps the H+ ion into the endosomal space. Then there is a protein which is M2 protein. M2, this is an ion channel that is present on the membrane of the envelope of the virus, that through which the proton or the H+ in the acidic endosome gets inside the virus. And that basically changes the the structure of the hemagglutinin, and it basically binds and fuses or flips its hydrophobic fusion peptide and binds with the membrane to basically release the content. And remember that one of the most important part, influenza is a negative strand RNA virus, and it has a segmented RNA virus. So just remember that M1 protein is entirely different, although mentioned here. Remember that this is nothing to do with M1 and M2. M2 is present on the surface of the membrane. This is a different protein entirely. So that's one of the important things. And here is the mechanism, what you are looking at. Here is the M2. This is the viral envelope. And the amantadine basically binds and blocks a couple of places on the of the M2 protein, so they cannot make any conformational change to allow the proton to enter into the virus. This is inside the virus, and this is inside the endosome. So this pathway is basically blocked. And sometimes we can just change that. And one of the most important part that a lot of people actually got tricked. Sometimes you can have a trick question. Remember that right here is an anti-influenza A virus drug. If everything I say is correct and just change this to influenza B, it will be incorrect. I know that this is a mean question, but as I'm saying it, it should not be mean. So just remember that it's not going to work for influenza B. If you see influenza B, amantadine. So just remember A for amantadine, B not amantadine. So even if the process I say is correct, if it is influenza B, that should not be your answer. So just those kind of questions that we can ask. All right, so after that we are looking at this oseltamivir or the Tamiflu. This is a neuraminidase inhibitor. And just a couple of questions from that we can ask which of the following is true, and that kind of question is just remember that this is designed to mimic a natural ligand, which is the sialic acid. You can see that it's binding here. The drug works during the exit or the budding of the influenza virus rather than the entry of the virus. So just remember that what happens that they are all stuck together during the exit from the cell, and that's how the neuraminidase basically works. So that's one of the important things. That if you just remember that, that's good enough. Also, if you just look at the top, you can just say that this is also influenza A. It doesn't work very well with the influenza B type. So even right here, the age-old amantadine or Semetrol, as well as the Tamiflu, both are influenza A drugs. All right, anti-HIV drugs. So just know that what they are. You do not have to know the details or the names of each one of them. Just remember that there are fusion inhibitors, then you have reverse transcriptase inhibitors, integrase inhibitors, protease inhibitors. These are some of the most important ones. And one of the things also remember that the hepatitis B virus, which is the HBV, which has a gapped genome that we talked about, entirely different thing. It's a gapped DNA genome, but it contains a reverse transcriptase in its life cycle, but it doesn't integrate its DNA into the host. And that's the reason that what happens that reverse transcriptase is reverse transcriptase, no matter whether it's a DNA virus, but the enzymes have a similar function even in different system, in the viral system. So what can happen that the RT drug or the anti-RT drug which is used for the HIV, that same drug can be used for hepatitis B virus as well. Is tenofovir, there is a specific drug. I probably just gave a question in the quiz as well, you probably will see. But there is an exact same drug that you can use for hepatitis B infection, as well as the same drug will work for an anti-RT in case of HIV. So just know that. And also along with these, you also have the nucleoside analogs, which is not shown. The nucleoside analogs, you know that how it works, that it would cause the chain termination. All right. So that's basically that nucleoside analog chain termination, but these are the details of those, really not required. Yeah, so these are some of the historic drugs, so definitely we need some respect for that, which is the ribavirin and the foscarnet. And what is the, and they are not prescribed anymore as such. In most of the cases, it's available in the market. You can find them, but they are not prescribed and hardly they are used. And if it is asked that what is the reason that they are not used, just remember that they are extremely toxic. It's a nucleoside analog prodrug, very toxic, cause bone and the renal toxicity for foscarnet. And these are some of the earliest known broad-spectrum antiviral and that treated many of the Ebola, Lassa fever, Hanta fever, respiratory syncytial virus infections as well. So very straightforward questions. All right, so after that, what we are looking at is that the favipiravir is basically a guanosine nucleoside, which is analog prodrug basically and active against many of the RNA viruses. And there is a cellular enzyme which is responsible for converting this prodrug to the drug producing the phosphorylated form. See that right here, this doesn't have any phosphate. If you add the phosphate right here to this nucleoside, before it crosses the plasma membrane, it cannot get in. So remember that, because it will just become polar. It's very difficult. So here it is a nonpolar molecule. Nucleoside can pass through the plasma membrane. And now in the cell, the AGPRT adds the phosphate and then makes the triphosphate with the other enzymes that can be incorporated, and the chain termination or lethal mutation can happen. So this is what you need to remember. It's a big name, but it is AGPRT, which is the hypoxanthine guanine phosphoribosyl transferase. This is the enzyme, which is a cellular enzyme, puts that phosphate on the favipiravir. So just again, just all you need is this, that mode of inhibition, but the question can be asked that which of the cellular enzyme can convert the prodrug to the phosphorylated form, basically. All right, so we are good. We're moving into right here. All right, so here is we talked about many times. You just probably got this one. Is the remdesivir is being used for the treatment marginally, and contain the proofreading enzyme. EXO-N and ENDO-U, you know that all of the coronaviruses contain that, and SARS-CoV-2 has no exception with that. So the efficacy of the drug is less than what is expected. It's still being used, but probably reduce the intensity of the disease probably 1 or 2 days, but very marginal. This is and we can ask that what is the reason for that? That why doesn't it work? Or the failure of the remdesivir as a cure. It really cannot cure the disease. It just can make the symptoms a little bit milder. And the reason for that is the presence of these exonuclease EXO-N and the endonuclease ENDO-U. If they're misincorporated, they can really remove that by this proofreading function of the RNA-dependent RNA polymerase complex. So that will do it. Oh, so this is the interesting part. I don't know whether you have seen in the news or not. So day before yesterday, as I said, that I changed this word. This is like if, when I presented this, it was if authorized, and it's authorized day before yesterday. So this is a molnupiravir from Merck, is the first antiviral. Marginal, don't think that it will be better than remdesivir, maybe marginally, but this is approved by FDA. But not this one yet. But this is probably going to be the game changer, we think, probably. So Paxlovid, because it is so much interest in that, that's why we can just ask that what it basically does. So here we talked about these material in our extra special topic mainly, but you know this, so I'm just explaining that how this does work. So the RNA viruses, all including the SARS-CoV-2, you know that there is this replicon 1A and then the replicon 1B, and in between them they have this pseudoknot structure and a stop codon. So the mRNA is all straight, just contains the rep A or rep 1B. Upon entry, it translates directly into a polyprotein 1A, and sometimes we talked about this, that there is a slippage of these ribosomes on a UAA track and then followed by a pseudoknot. And sometimes that it slips out of frame, -1, and when that happens, it cannot recognize the stop codon and it does synthesize the whole thing, which is a polyprotein AB. So you can see that polyprotein 1A, 1AB. Now, if you look at that NSP5, which is right here, NSP5, this is the main protease, also known as a 3CL protease. And the cleavage site where it cuts are these arrows. Whereas I did not produce that, NSP3 is another protease, and those NSP3 cuts at these points. So where you see these dots right here, so it will cut 1, 2, 3, and 4. 1, 2, 3, and 4. But 5 through 16, all these different proteins are made from the polyprotein by the digestion of the NSP5. So that's one of the important things, that just remember that right here, as I said, that the protease cleavage is necessary to produce the viral NS5, oh, not 55, I'm sorry. Right here. NS5, non-structural protein 5 through non-structural protein 16. And if you block this NSP5 with the protease inhibitor, which is the Paxlovid, then this cleavage will not happen. So basically, upon infection, the mRNA is translated into the polyprotein, and the polyprotein can make 1 through 5, but it will not be able to make any of these. And remember that right here, these are the ones which make the RNA-dependent RNA polymerase. So right here, 7, 8, 9, 10, these are of the methyltransferases and RNA-dependent RNA polymerase. See that these are all making the holoenzyme of the RNA-dependent RNA polymerase. And if these are not cleaved and assembled together, there is no replication. So that's the mechanism that just inhibiting the protease can do this. So just know that how this basically works. So here, the most important part is that underline again that the viral protein NS5 through NS16 will not be produced if NS5 is basically blocked. So that's your take-home message. All right, so let's go to the last one. How much time we have? Yeah, we have enough time. So just talking about that when origin, about 450 million years ago, just straightforward when you think the virus basically originated. And then the different, just know the definition of the cellular regression escape gene and the primordial or the virus. This is also known as a virus first hypothesis. So just know that what the hypotheses are. We're just going to give you the definition and ask what it is, or give you the name, just the either way. So just the definition of each one of them, and that will do it. Let's see right here. So we're just skipping a lot of them, and we are stopping right here. Yeah. So here I just changed this slide a little bit. This side, this was not there. And the question is that if we just ask, is that which of the following is a driver of the viral evolution? And this was explained in here, and it was in the notes, but here, just remember that these are the overview of the evolution. Is a large number of progeny and large number of mutants, as you can easily see, and the quasi-species effect, and the selection of a fit population of the viruses, as you can easily see. So large population passages means that progeny viruses increase replicative mutations, improve replication, mutants replicates, and the quasi-species. They have all these genetic diversities that basically drive the viral evolution. And Another question that can be asked, this is an extra slide. This was not there in your stuff, so I just pushed it aside, that where from it comes from, that how the rapid evolution of SARS-CoV-2 is happening. Just remember that these are the things that are coming from this. Basically, there's a large pool of virus and many susceptible hosts that the virus can infect. High viral load, many different mutations accumulating quasi-species are constantly generated, which is from this slide. Some mutated quasi-species become more fit for survival, for example, being more contagious. And as a human population are taking measure against the viral spread, vaccination, masks, social distancing, the viral quasi-species to counter the measures are selected, such as being more contagious. When it is not be able to connect the one which has a mutation which can basically cross even in the, under that situation is basically selected. So this is a very important slide, and in the present context as well. Now looking at some of the bottlenecks that how the genetic bottlenecks that say for an example you constructed if I just give you a just kind of a question that you constructed a virus and you can see that after a few passages the multiplicity of the infection actually dropped dramatically, and what could be the cause. So when you do those, usually you plaque purify, which is a genetic bottleneck, and that can reduce the fitness of the virus, and you can basically see that kind of problem, that the virus is becoming less and less infectious. All right, so here some of the questions that we can just ask is that how the viruses can escape the Muller's ratchet, like accumulating a large amount of accumulating constant the constant mutations and how they escape. Remember that, how to escape the Muller ratchet. Remember that infections by a large, diverse quasi-species population of the virus is the key, so that the virus remains basically fit. On the other hand, here you can see that what are the when you make these issues, the small droplets of suspended virus aerosolic transmission, activation of latent virus from a limited population of cells, small volume of inoculum, these are all bottlenecks. But this is how the bottlenecks is surpassed. Infection by a large, diverse quasi-species population of virus. This is how it escapes these genetic bottlenecks. All right. So here is a very simple slide. There is nothing specific, just know the definition of these different types of viruses that what we talk about. That will say, for example, what kind of viral host interaction is observed when the virus is jumping from a group of experienced host to the naive host right here. So that's experienced host to the naive population that's evolving. Say on the dead end one-way passage, which is also called a spillover. So sometimes this can happen from a zoonotic source or an animal. So that's basically just usually from an animal. Just remember that usually a one-way passage spillover non-efficient transmission is usually from an animal, which we call as a zoonotic, Z-O-O, then notic, N-O-T-I-C, and zoonotic transmission. So just the basic definition of these. Just informational, I'm just not going to ask many questions about the information. Just know that what was the Hendra virus and the facts, basically. Australia, racehorses, just a random question can be asked from here as well. No, this is not, yeah, so don't worry about that. Yeah, so here just everybody talks about Ebola. Some of the facts know that where it started, basically, that's the fact, and then a couple of other facts yeah, so here basically that if we just give you that which of the following virus actually started in 1976 in the Democratic Republic of Congo and have more than 80% fatality or mortality, remember that is basically we are talking about the Ebola virus, basically. Same question. All right, so here just the zoonotic reservoir for the coronaviruses. We know the palm civet cats and dromedary camels for the MARS, so know those. And as of now, we do not know for the SARS-CoV-2. It's just the basic idea that what are those. So yeah, just the host, so just remember those. And so this is just a historic perspective that how it was traced, the origin of the SARS-CoV-2, just exactly how the underlying part is exactly how it is. Human sera collected, and they were found that they have a high amount of SARS-CoV-2 antibodies in the control group, and then it was just found that the meat market is basically thing. So here is the MERS. Just remember that it uses a different receptor, and you can be asked a kind of a critical thinking question regarding those. That, say for an example, we can say that we have delivered a virus using a liposome into the cell, but after the virus replicates in that cell, it doesn't infect any neighboring cell. And what could be the reason? One of the reasons it could be that it uses a different receptor, and that cell didn't have a receptor. So just make sure that you can just rationalize those questions that way. All right. So is that all? Pretty much. Looks like 9:13. Yeah, that's it pretty much, I guess. Any questions? And again, as I said, is like look at your quiz questions as well. And as I said, that you will see a lot of these will correspond to some of the relevant quiz questions. So take a look at those, and that will basically help a lot with this as well. So here is one question someone asked me. This is not related to the exam, just spending a single time. Is they asked me, is like you were saying yesterday as per the thing, that there are antibodies against this region, but this is the region which is the receptor binding domain of the spike protein. And basically, if you are not having an antibody, how you are basically preventing or neutralizing the infection? So very good question. So remember that there are two things. First of all, the fusion peptide is at the interface. So if you have an antibody in here, even if it binds to the receptor, that antibody will block that fusion peptide, and basically it just doesn't allow the fusion to happen. That is one of the reasons. And the fusion peptide is in this region, remember that. On the other hand, another important part is that it works in a very different way. As soon as the antibody is coated, remember that this is the most important part. As soon as the antibody coats the surface of the spike protein, even if it's not on the receptor binding domain, immediately what happens that there are these complement based system that will come over here, and the virus will basically aggregate in the large amount, and the macrophages come in and they engulf this, which is called as an opsonization, basically. So you will form this lattice of viruses connected by these antibodies, because one antibody arm can bind to this one, another can bind to the next virus, and they form this lattice, and they are basically neutralized that way. So even if they don't bind to the receptor binding domain, then still the vaccines should be pretty effective as well. And of course, I mean, like if you have it from all over, that's better. Efficacy will probably be dropped, but hopefully we'll keep an eye. All right. So any other questions? So all the best for the exam, and hopefully it will not be stressful. You can probably see it now because I just published that in there. So it will be open Wednesday all the way through Friday. You can take it anytime. It will be the same way. It's not open note, open books. It's the same kind. So if you study, and you should not have any problem, no surprises as such. But do the quiz question. All right. So all the best for the finals week. And then happy holidays, and then happy New Year. And if you just don't be a stranger if you see me in the campus. And whatever you celebrate, Merry Christmas, Happy Kwanzaa, Happy Hanukkah, whatever you celebrate, and enjoy your holidays. All right, it was a pleasure.